Buenos días, gracias por estar en el segundo día de los seminarios de Fronteras como parte de del aniversario 95 de nuestro instituto. Recuerden que mañana es el gran día, pero hoy también lo va a ser. Entonces, vamos a empezar. Hoy vamos a tener otras tres charlas de nuestras ponentes y nuestros ponentes que nos visitan de fuera de nuestra ciudad, incluso fuera de nuestro país. Voy a dar unos anuncios para iniciar, que son de seguridad. El primero es, recuerden, si suena la alerta sísmica o cualquier alerta en nuestro instituto, la mitad de aquí para acá, por favor, evacuar en esta puerta y la otra mitad en la otra, que es básicamente la más cercana, para reunirnos en el punto señalado con verde aquí en el estacionamiento. Por favor, celulares, si pueden ponerlos de una vez en silencio o apagarlos y liberar espacios, sobre todo los pasillos y las escaleras en caso de tener que salir. Sin más, voy a dar inicio a la presentación de nuestra primera ponente del día de hoy. Vamos a empezar con zoología, lo cual es muy, en mi caso, me interesa aún más. Y es la profesora Andrea Waeschenbach, que nos visita del Natural History Museum de London. Ella está trabajando actualmente en el departamento de Life Sciences. Y voy a leer brevemente un poquito de sus intereses en investigación, así como parte de sus publicaciones recientes. La doctora Andrea es sistemata molecular con un interés muy particular en dos grupos, que son los briozoos y los platelmintos, y está interesada particularmente cómo el uso de las filogenias le permite estudiar los patrones de diversificación y los procesos subyacentes. Su investigación como tal también está dirigida hacia los componentes morfológicos clave y los atributos o rasgos de las especies para comprender su importancia evolutiva y muy particularmente la interacción que existe entre los parásitos y los hospederos. Ha utilizado muchos datos genómicos, particularmente genomas mitocondriales, utilizando técnicas de nueva generación o de última generación para resolver preguntas evolutivas. Como un comentario adicional, también ella está muy interesada, y esto es bastante atractivo también, en la identificación molecular de todas las aves que son víctimas de las colisiones con aviones. Ella obtuvo su doctorado también en el Reino Unido, así como su licenciatura en el mismo país, y tiene un sinfín de publicaciones, no solamente en revistas dirigidas a parasitología, sino también en sistemática molecular, incluso en paleobiología. Sin más, quiero darle la bienvenida a la doctora Andrea Waeschenbach y a la gente que nos ve en YouTube. También les recordamos que pueden empezar a hacer sus comentarios y preguntas para tomarlas al final del seminario. Muchas gracias, doctor.
Thank you, Lazarus. First, I'd like to thank the organizers for the generous offer to invite me here. This is my first time in Mexico, and I'm having a great time. So thank you very much. So you might be wondering why I'm showing you a picture of fish, but I'd like to draw your attention to this part. So all these orange branches that you can see here, they are bryozoans. This green colony here is also a bryozoan, and much of the beige turf is bryozoans. So they are really important to generate a complex habitat for other organisms to live on, and they are often overlooked. Looking at the program, I thought there would be a lot of botanists in the audience, so I thought I would start off with I would start with some herbarium specimens of bryozoans. So historically, bryozoans were sometimes placed in herbaria because they were misidentified as seaweeds. So we have here a type specimen of Alcinidium diaphanum, which was originally described as Ulva diaphana, but it's actually a bryozoan. And here we have Flusterfoliacea, which was labeled by botanist Samuel Dale as a fucoid alga, but it's a bryozoan. And I just wanted to start off with this quotation by Carl Simpson and Jeremy Jackson. Bryozoans are neglected children of the sea floor, and that's true in many ways. And I'm just hoping to, over the next hour, to shine the spotlight on bryozoans. So just to give you an overview, I'm going to give you an introduction to bryozoans, and then I'm going to give you some insights from molecular phylogenies, and specifically looking at some convergences of morphological characters, looking at the phylogenetic distribution of reproductive traits in a particular group of bryozoans, and also giving an example how we use phylogenies for taxonomic work. And then I'm going to move on to talk about macroevolution and also finish off with some notes on genome architecture. So to introduce bryozoans, these are, apart from some exceptions, colonial animals. So they're made up of individual modules called zooids. And you can see these are the feeding zooids that you can see here. And they're feeding with a structure called a lophophore, which is a ciliated crown. We find bryozoans throughout all aquatic habitats, marine, brackish, and freshwater. We also find them all the way from the intertidal to the deep sea. I think the deepest record is about 9,000 meters. We find them all the way from the polar regions to the tropics. Most bryozoans produce a calcium carbonate skeleton, but some are also uncalcified. And there's about 6,000 extant known species. Of course, we're discovering new species all the time. So just briefly, it's a colonial animal. So what they produce a larvae, planktonic larvae, which then settle on a suitable substrate, metamorphose in the founding zooid of the colony called the ancestrula, which then by asexual budding gives rise to the rest of the colony. Now, the different parts of the colony, the different modules or zooids, they can perform different functions and they can look very differently. So we have polymorphism of zooids, which perform division of labor. So here we have an example where we have a feeding zooid. So this is obviously, the lophophore is not there because it's dead. Here we have an avicularium, which is a defensive zooid. So you can see that little pivot bar here. So it would, because we bleached these specimens, and this there was a cuticular mandible that was sitting on this, and it's like a little flap that flaps open and shut, and it can trap predators and things that might want to settle on the colony. And then we have modules which are involved in reproduction. So this is a developing ovicell, which is a brood chamber. Here we have an example. This is a different kind of bryozoan. So here, the larger holes that you can see here, those are the feeding zooids. And in between, we have what's called kinozooids, which are the building blocks. So they don't have a lophophore. They're not feeding. They're basically just there to separate the feeding zooids from one another so the lophophore can pop out to feed. In terms of colony forms, there's a huge diversity. This is a nice catch that we got in South Africa. And this is before they go into ethanol, so we can still see the beautiful color. So we have different kinds of colony forms here, lots of erect forms. This is like a little miniature tree. Here we have an encrusting bryozoans that grows on a mussel shell. We also have some free-living species. So this is Cupuladria biporosa from the Isthmus of Panama. And in this species, so you can see these little hair-like structures. So those are called vibracular, which are essentially, if you remember back, the defensive zooid with a mandible. This is a modified defensive zooid in which the mandible has been extended. So it's almost like a whip. And in this case, it uses these structures for locomotion. So these live on sandy bottoms. So they can move very, very slowly across the sediment. We also have one pelagic species, Alcinidum pelagospherum, which occurs in the Southern Ocean. And I mentioned we have deep sea taxa. I just wanted to draw your attention to this zone here, the Clarion-Clipperton Fracture Zone in the Pacific. Some of you might have heard about this area. It's a vast abyssal plain, and it's covered in these polymetallic nodules, which have been earmarked for deep sea mining, basically to get all the minerals for our electronic devices and batteries, etc. It's a very destructive way of mining. Huge machineries go down there. It's a very stable environment, the deep sea. So having something like that go in there, it's really, it destroys the habitat. And not only the habitat where they actually are mining, but it produces very large sediment plumes, which go over kilometers and suffocate marine life further down in neighboring areas. But crucially, as you're mining these nodules, which have taken 2 to 3 million years to form, you're actually removing the substrate for the marine life down there. And these are some examples of bryozoans that have been found on these nodules. This is some work from my Russian colleague, Andrea, who's been working on the Russian sector of the Clarion-Clipperton fracture zone. And what's interesting, bryozoans in this area, they often have stilts. So they have rhizoids. So it's to elevate them from the substrate. Because it's quite a fine silty habitat. Just to briefly go through the major lineages, we have the freshwater bryozoans in the class Phalactelimata. It's the least diverse of the bryozoans. They are all uncalcified, so they have a poor fossil record, and they're highly clonal. So they produce these resting stages called statoblasts, which can be used for dispersal. So they're propagules, but they're clonally produced by the colony. Then we have the Cyclostomatida, which is the only surviving order in the class Stenolimata. These are entirely marine, they're all calcified, and there's about 550 extant species. And then moving on to the class Gymnolimata, for which we have 2 orders, the Tinostomatida, which occur in marine, brackish, and freshwater habitats. There's about 320 extant species, and they're uncalcified, and it's actually quite nice because you can see the internal features of the zooid. So we have here, this is the lophophore, which is retracted. You've got the esophagus. Here we have a gizzard. So not all bryozoans have a gizzard, but it's in this group of tenostomes, we have a gizzard, and it crunches any kind of hard food. Then we have an intestine, a U-shaped intestine, and then the anus pops out here, outside of the lophophore. The other thing to note here is this tissue strand. It's called a funiculus, which connects the different zooids in the colony with one another. And then we have the superstars of the bryozoans, the Cheilostomatida. They're the most speciose group today, with about 5,000 extant species. They're entirely marine, they're all calcified, and they display most of the polymorphism that we see in the group. So why are we studying bryozoans? Well, I already mentioned that they are important habitat constructors. These are just some images to emphasize that point. So we have, this is a bryozoan colony. There's another bryozoan colony that lives on that colony. More bryozoans here. These are bryozoans. So they are really, they really enhance the local ecosystem. This is a bryozoan that grows in the English Channel. So we don't even need to go to the tropics to find them. The common name is Ross coral. Of course, it's not a coral, but of course, people like to give common names which don't always make sense. They can grow up to maybe 2 meters in diameter, but of course, with trawling and fishing, they get easily destroyed. And then here we have Cinctipora elegans, which occurs off the coast of New Zealand on the Otago Shelf. We have these bryozoan meadows. It's like a turf of bryozoans, which are really important ecologically. We also have invasive species. This is a species, Amethia verticillata, which supposedly originally comes from Italy, but has now got a cosmopolitan distribution, and was most recently also recorded from Oaxaca. Bryozoans also can be sources of biomedical compounds. So this is the most famous example. This is bryostatin. It's a macrocyclic lactone for any chemists in the audience. It's produced by an endosymbiotic bacteria that lives in this species, Bugula neretina. And it has some apoptotic properties, which are anti-cancer and also memory-enhancing properties. But there are many, many compounds that have been isolated from bryozoans that have potential medical applications. They're also important biofiling organisms. This is my colleague, Tim Wood, who has a consultancy in the US. He works around freshwater bryozoans, and he often gets called out by various companies, whether it's drinking water companies because their pipes have been blocked, or wastewater treatment companies. of course, crucially, they can also grow in the cooling water inlet pipes of nuclear power stations. And they are hosts of parasites, so they're definitive hosts for myxozoan parasites, and they can cause proliferative kidney disease in salmonids, which can have important economic impacts in fish farms. They also have a good fossil record, and this is just to illustrate very broadly the kind of bryozoans that we see since their origin. So there's still some controversy whether they originate in the Cambrian or the Ordovician. This is only going back to the Ordovician here, but so we're seeing up until the end of the Paleozoic, the group that are dominating are in the class Stenolimata. So it's only one of these, the Cyclostomatida that I mentioned earlier, that made it past the end of Permian mass extinction event, which happened here and diversified here. We have the Tinostomatida, so because these are uncalcified, so the fossil record is a bit patchy and not so reliable in this part. And then we have the Chylostomatida, which originated in the Jurassic, but really only took off in the Cretaceous. Just to mention here, you actually have in Mexico, you've got some really good Paleozoic outcrops. And this is an example of the Ixtatepec Formation in Oaxaca. And this is Sergio Gonzalez Mora, who has done some intensive work on this formation, looking at bryozoans. And he found, so the dominant group are the Fenestrata in the order Fenestrata and the Cryptostomata. So just a little note of what the fossils that we have in Mexico. So I'm now going to move on to insights from molecular phylogenies. And I'm going to start off with the Cyclostomatida. And I just wanted to quote Arthur Waters here in 1884. I have known that the Cyclostomata must shortly be dealt with and have looked forward to this with a feeling almost of dismay because they possess so few characters that can be used for the purpose of determination. Now, forward 140 years, this hasn't really changed. So we have these 5 suborders, and they display a lot of different colony morphologies. So there's a lot of diversity amongst them. The Cinctiporidae is a family that hasn't really been assigned to any of the suborders yet. It's an unusual one because it has very, very large zooids, larger than any of the Cyclostomatida. And what they don't have any brood chambers. So they're called gonozoids, which you can see here in this species. It's centrally organized. Here you can see a flask-shaped gonozoid. So this is where the embryos are being incubated in this group. Now, historically, people have used this character, the way that the skeletal wall is constructed, as an important character in terms of the classification of this group. So we have 2 different types of wall constructions. We have the free-walled constructions, which is actually, so those are the green taxa. And we have a skeleton here, which is sandwiched between 2 epithelia and 2 pseudocoels, then another epithelium, and then a cuticle, so the outside is here. So quite a complex arrangement of the skeletal wall. And then we have the free-walled ones, which are here in orange, and they have just 1 layer of epithelium, the skeleton, the cuticle, and then the outside. And then we have the Serioporina, which have free-walled autozoids, which are the feeding zooids, and fixed-walled gonozoids, so a mixture. Now, a few years ago, so this is my paleontology colleague, Paul Taylor, who used to work at the Natural History Museum. He's now retired, but we still work together. So a few years ago, he said to me, look, wouldn't it be great if we produced a phylogeny of cyclostomes? So I said, yeah, let's do it. This is the phylogeny that we produced in this paper. So we've got 3 major clades. The Cinctipora, the ones with the large zooids, couldn't have been placed unambiguously. But what's crucial is that I mapped here the different kind of skeletal wall constructions, and they are all over the place. So when I first showed this phylogeny to Paul, who's been working on this group for much of his career, there was a long moment of silence for him to digest this result. And we continue to sequence in this group, and every time we sequence something, we get another surprise. So there's a lot of convergence of morphologies in this group. Then we come to, this is another Cyclostomatida in the order Cancellata. So this is a CT scan. So you can see here, so we're going now towards the back of the colony. So all the autozooids are on the frontal surface. You can see here these bulbous structures. These are the gonozoids. You've got another one here. And what's interesting, you see that line here and this line here. This is another bryozoan. This is actually a chylostome growing on this colony. This colony is about this big in real life. And then you have another chylostome growing here. And as it moves around, there's another cyclostome actually living here on the colony. So they have their own like little micro universes in the colonies themselves. Now, this is an example. So these are Honera robusta and Honera foliacea. These are species that occur in southern Australia and New Zealand. And people have always kind of thought, okay, there's only 2 species. But when we looked at it with a molecular phylogeny, so first of all, we've got a new genus here. And within this Foliacea group, we actually find that the Australian and the New Zealand specimens are separate species. But it's really in the Robusta clade that we find most of the hidden diversity that hadn't been recognized before. So 5 new species. Here we have the Robusta sensu stricto. Okay, moving on in the tree, we have the Tinostomatida. This is a study by Sebastian Decker and colleagues. And this was published earlier this year. And he found 3 major clades, A, B, and C. And what we are doing now, we've been expanding this phylogeny. And this was kickstarted by master's student Sichun Zhu. And so we're actually in the process of almost submitting this paper now. And the data that we're using for this are genome skimming data. So it's mitochondrial genomes and nuclear ribosomal operon genes, so principally 18S and 28S. And for this paper, we got 14 complete mitogenomes and 18 almost complete mitogenomes. And we sampled across a large diversity of tenostomes here from specimens from all around the world. Again, we found clades A, B, and C. And we also found several new species. Several new species in this study, as well as we're doing some reclassification as well. So this is a simplified version of that tree. So one of the things is, how can we define clades A, B, and C? Are there any kind of characters? Morphology doesn't really do it. So we looked at some kind of ecological traits, freshwater. What kind of habitats do they live in? So the green are the freshwater taxa. Then we have in blue the marine taxa. The only really signal is that in this clade we have some brackish species, but otherwise the marine and the freshwater taxa are kind of intermingled. Similarly, if we look at lifestyles, so the tenostomes have a diversity of lifestyles. So the turquoise ones are the epibenthic ones, so they just live on the substrate. Then we have some which live inside calcareous substrates, such as mollusk shells. So they bore into the mollusk shells. Then we have some wood-boring taxa in orange. This one species that lives inside polychaete tubes. We have some pelagic taxa that I mentioned earlier here, and also a free-living taxon. So there's no pattern here, and I can continue. And it's really difficult to find traits that define those clades. Moving on in the tree now, coming to the Cylostomatida. And in this study, we wanted to dissect the transitions of various reproductive strategies and brood incubations in the chylostomes. And this was led by master student Heather Grant, and we also had involved Andrea Ostrowski, who's the guru of bryozoan reproductive biology. So with bryozoans, we have some species which are zygote spawners, so they are non-brooders. So basically they release fertilized eggs through this intertentacular organ here. So you've got some more fertilized eggs waiting here. So what happens with these eggs get released and they develop into long-living planktotrophic larvae. So they can travel quite a long distance before they settle. And then we have the brooders. So here we have an ovicell, which is a brood chamber in which the embryos are being incubated. And the larvae that are being released are short-lived, non-feeding larvae. Now, in addition to the ovicells, we're also seeing a lot of different types of brood incubation methods in this group. A huge diversity. So we've got internal brooding. We've got acanthostegal brood chambers where you can see here, these are the spines that are overarching the membranous frontal wall of the organism, producing a brood chamber. We have external membranous sacs. We also have, so I mentioned the kenozoids earlier, the building blocks. So in this case, the kenozoids are producing a brood chamber in this species. We also have a brood chamber which is produced by the maternal zooid as frontal outgrowths. And then we have, again, a kenozoidal arrangement. It's called a synoecium, in which we have the kenozoids are like miniature adult zooids. And so this is actually broken up. But they basically, the miniature zooids, these kenozoids, they produce a brood chamber as they, form a brood chamber like that. So we did we produced a genome skimming phylogeny for this group, and we did some ancestral character estimation. So we have, this is the base of the Chilostomatida. And we see that although it's not entirely unambiguous, but there's some evidence that the ancestral state of the chilostomes is a zygote spawning state. And within that, we have the various brooding taxa nesting. So we have, I mentioned external membranous sacs. There's actually 4 different types of those distributed throughout. We also have, so we've got acanthostegal brood chambers up here and the kenozoidal bilobate brood chamber here. And then we have a whole clade of brooders. So we've got the synoecium here, and then we have Steganoporella and Salamoporella here. And it seems that this kind of state has evolved from the internal brood sac, which is actually contrary to what my colleague Andrea Ostrowski believes. He thinks it's the reverse, that typically these brood chambers go inside and become internal sacs. So it was a bit of a surprise for him to see this result. And then we have the so the ovicells, which what I've shown you earlier, and they form a monophyletic group down here. But overall, we can say that a lot of these different brood incubation methods have evolved from a zygote spawning ancestral state, most likely. So coming now to macroevolution. So in terms of species diversity through time, there's a general consensus that there are 2 different forces that produce these changes in species diversity through time. We've got the abiotic forces from the physical environment, and these are typically unpredictable and large-scale events, which have caused the mass extinction events that we've seen in the past. And we have the biotic forces, which are happening at a more local level, at the ecological level, with the interactions such as competition and predation. But both of these forces supposedly have an effect on species diversity through time. Now, we can estimate extinction and speciation rates either from fossil data, so we can either do direct counts of fossils, or we can model fossil occurrence data So there's a paleobiology database which hosts a huge amount of fossil occurrence data. Or we can use molecular phylogenies, time-calibrated phylogenies. So we have a lineage to time plot here where the slope of this curve indicates, you know, if there's a lot of lineages occurring in a short period of time, the slope is a lot steeper than, for example, here. But of course, the problem with the time-calibrated phylogenies, they typically only include living species, so it's really difficult to estimate extinction rates from those trees. Just briefly revisiting this figure, so I'm going to be talking about the diversification dynamics of this group, the Chilostomatidae. This project, there's a lot of people have been involved in this project as well, because we had to produce a molecular phylogeny, so a lot of people have been involved with getting specimens for us or helping us to collect specimens. But I just thought I'd mention some core people here. So the postdocs up here. So we've got Helen Jenkins and Rachel Graham, who were dealing with the molecular data, and Farideh Mourek and Paul Taylor, who were dealing with the fossil analysis. It's actually Paul Taylor, who was my postdoc supervisor for a period of time, became my postdoc when he retired, which is which is very enjoyable. And then we also have two people here, Dennis Gordon from NIWA in New Zealand and Leandro Vieira from the University of Pernambuco in Brazil, who've been really been involved throughout the project quite heavily. And these were our funders, the Leverhulme Trust, for this work. So we wanted to basically produce an updated estimate of extinction and origination rates, and basically providing an update of what was published previously in these papers. Some of you might be familiar with Daniele Silvestro. He wrote the program Pyrate. Pyrate is a Bayesian program which takes fossil occurrence data and produces speciation rates, extinction rate, and preservation rate estimates. And so this is really the first time that a Bayesian method has been applied to the Chilostome bryozoan fossil record. So, so what we did, so in this paper, we actually analyzed a bunch of different data sets. I'm just showing you the results of this particular data set, which is basically the paleobiology database occurrence data, but we had to clean it up because there were lots of errors and synonymies. So you can't just download and analyze. So you do have to triage the data a little bit. And then we augmented it with published and unpublished occurrences. So what you can see here is so we have origination rates, extinction rates, and diversification rates here. Here you can see there's an asterisk here, and asterisk here, and here. Those are the significant rate changes that we observed. So you can see here there's a long macroevolutionary lag from their origin in the Jurassic to the mid-Cretaceous and the Albian. Now something happened here, and that is the origin of ovicells. It's the origin of brooding. And my colleague Paul Taylor, in his 1988 paper, he hypothesized that the origin of brooding, because the larvae that are being produced, they are non-feeding, so they don't travel so far. So there's less gene flow between distant populations. So you're generating a more genetically heterogeneous landscape, which might be more, which might foster more speciation. So that happened at this moment here. And we can actually see on this plot here, this coincides with a peak. So you've got the Albian here, and then further on, the Cinnamanian and the Turonian here. So that it basically correlates to this part. The other thing that happened here is we have the origin of frontal shields. So the very early bryozoans, they didn't have any frontal calcification. They just had a membranous frontal. In fact, we still have some of those today, but of course they're very vulnerable to predation. So the origin of the frontal shield supposedly also been one of the key innovations that helped the group diversify. And then, of course, we have the KPG mass extinction event with the Chicxulub impact. which caused the mass extinction at that moment here. And we see a decline in diversity following this impact. And we also see a big extinction peak at that moment. Then we get, basically, it recovers again. And so we had these frontal shields, but we have two more frontal shields that have originated. And in this paper by Cheetah in 1971, he considers this the second evolutionary episode. So this return of diversity is probably largely driven by species which have a frontal wall. And we can also see an increase here, a significant increase here. We also did a correlation analysis of the origination rates and the extinction rates with some abiotic factors. So we've got sea surface temperature and sea level. So what we found was that origination rate correlated positively with sea surface temperature. So, well, why might that be? Well, the warmer, the faster the mutation rates are, the shorter the generation times are, the faster metabolisms, and that means shorter larval durations. And I already mentioned that earlier, the shorter larval durations means less dispersal and less gene flow, all of which might have had an effect on a higher speciation rate. And then we also have a negative correlation with extinction rate with sea level. So why might that be? Well, as sea levels are rising and they're flooding the continental shelves, you're actually expanding the ecospace and increase habitat complexity. And also, as you are flooding these regions, you are releasing nutrients from the soil and the rocks, which can lead to increased phytoplankton growth. And of course, for suspension feeders like bryozoans, that's good news. So I have to say these are very broad brush analyses, but it's just nice to, for a first start, to look at the fossil record of this group with a Bayesian method. And of course, what we also want to do, so we've also been working on a molecular phylogeny, and we want to time calibrate it. So we have here, this is a paper by Russell Orr et al., published in 2022. And so they have a very well-sampled phylogeny. And the only thing is here that the origin of the chylostomes, so that's the molecular estimate here, so it's this part of it's this node basically, is very much older than the first fossil record, which is 160 million years ago. So there's a huge gap between the earliest fossil and the molecular estimate. So the method that they used in their paper is they use node calibrations and they used a fixed topology in the program MCMC tree. What we would like to do is, so this is work in progress, is to use a total evidence approach where you add the fossils into the phylogeny as terminals, and you can essentially use as many fossils as you like, as long as they fit nicely into the phylogeny. But for this, we need to produce a morphological matrix, because that's how you add the fossils into the phylogeny. So you've got a molecular matrix, and then you append a morphological matrix for the recent taxa that you've sequenced, as well as for all the fossils that you want to include. And this is a nice illustration of the characters that we can score. So this is a fossil and this is a recent taxon. This is actually a particularly nice example, I have to say. But you can see that the skeletal characters are pretty much equivalent. So that's a nice thing about bryozoans. I have to say, I talked with Susanna about that yesterday at lunch. Building a morphological matrix is not for the faint-hearted. It's a very labor-intensive process, and these are the key people that have been involved in this process. And so where we are now, we've got in the matrix 419 species, so 276 recent molecular taxa and 143 fossil taxa, and we've got 187 discrete characters. Now, of course, it's too tempting, even though we have only an interim matrix. It's nice to run some analyses, right? You want to explore the data set. So we've got our genome skimming phylogeny, and then we have our, so I ran a morphological phylogeny here. So what I did here, this is obviously a much bigger tree than I can show here, but I cherry-picked fossils which actually fitted quite nicely in the phylogeny, that made sense. And then I included them in a total evidence analysis. So you can see all the red lines, those are the fossils all distributed throughout the tree. We had to constrain the topology quite a bit to also help the fossils to find their way. But what is quite encouraging is that the origin of the chylostomes is much more in line with what we know from the fossil record. The only thing is that the root is quite a bit too young, so we need, obviously this still needs some work, but I'm hopeful that this is going to be a productive method. Okay, just to finish off on some notes on genome architecture. So there are, following yesterday's talks, Jim's talk, you know, lots of genomes out there. We don't have the same situation in bryozoans. There's very, very few genomes. So we have the Darwin Tree of Life that Jim already mentioned yesterday. It's a UK initiative which plans to sequence 70,000 eukaryote genomes in the UK and Ireland. Great Britain and Ireland. And so this is one of the outputs from the Darwin Tree of Life. So, you know, there's only I think this is a genome that already existed. It's the freshwater species here. So there's only 4 new genomes that are analyzed here. But what we can see here, so we've got the different chromosomes up here. And what we can see is that the chromosome architecture, the chromosome structure is quite conserved within the chylostome. So these are all chylostome bryozoans, but it's very, very different to the freshwater bryozoan. And if we now put this into a wider context of bilaterian animals, so we've got a chordate here, we've got a mollusk here, and a nemertean here, and here we have the chylostomes, and here the freshwater bryozoan. And we can see, although these taxa are really quite distantly related, there's quite a lot of conservation in the chromosome structure. However, both chylostomes and the freshwater bryozoans are showing very divergent structures of the chromosomes. So this is kind of interesting, because what does that mean for the gene expression, you know, in this group? How does it actually work? Is there something different? It's actually maybe because we've got a lot of genome people here, being actually interesting to see whether there's some ideas of what, you know. I think this could be a good organism to explore those kind of mechanisms a bit further. So that was the nuclear genome. Now moving on to the mitochondrial genome, we get huge gene order rearrangements in this group. So every arrow, a different color is a different gene, and this is only a snapshot of what we see. Pretty much almost every species has a different gene order. And the other thing that we found is, so we didn't find this in all the mitogenomes, but maybe in 10 of the ones that we analyzed so far, we find introns in the protein coding genes. And that's highly unusual in bilaterian animals. I know it's common in plants and fungi, very unusual. So this is like, so when we annotated the mitogenes, we looked, what's going on? Why are the open reading frames of the protein coding genes interrupted? So we had to look at this in more detail, and we found that there are probably group 2 introns, which are self-splicing ribosomes. So this is not my topic. So we had to engage with this because that's what we found. So we found, so that's probably most likely because we've got the blue, so all the pink ones are the introns. We've got the blue bars here, which are the conserved domains in the ribozymes. And we also have intron encoding proteins, reverse transcriptase, and immature maturase. So this was an unusual finding. So we found this in this paper. We report that in 4 species. So in 2 of them, we could say they're probably group 2 introns. But in 2 other species where there were even more introns, it's a slightly different scenario again. So the intron encoding proteins, they're not actually in a protein, they are outside proteins. They're still there, but still it's different. And sometimes, lots of introns, sometimes 3 introns per protein coding gene, very, very unusual. So I've covered a lot of ground, but I hope that I've shown you that bryozoans display a huge amount of diversity of morphologies, many of which evolve convergently, lots of different lifestyles, reproductive strategies. And in terms of the macroevolution, to tease out the abiotic and biotic factors and the contributions from the key innovations on the diversification dynamics, we really need to look at this in a more granular level. Yes, and there's highly rearranged nuclear mitochondrial genome architecture in this group. And that just leaves me to thank you for listening, and thank you for IB UNAM for inviting me. Thank you very much.
Thank you very much, Professor Waeschenbach. So we have time for enough time for the question. But before that, I want to say that a couple of days ago, Professor Waeschenbach was teaching a workshop about genome skimming data. Thank you very much. This is very helpful for our students, for our grad students. So thank you so much.
Could you elaborate a little bit about the phylogenetic position of bryozoans? Because you went straight to them, but it calls my attention. They're colonial.
Yes.
But they're lophophorates, right? And
yes.
Other lophophorates are not colonial.
So we have there's one phoronid species which is colonial. So I've got this slide up here. So you're talking about the tree of life. Where do they fit on the tree of life?
Yes, and specifically the colonial condition, is it a derived condition? Because when we think of colonial organisms, we often think of, I don't know, coelentrates and they're far away. They're
absolutely, totally.
They're diploblastic, and now we're a little bit higher on the tree.
Exactly. So I had this slide in there, but I was worried I was running out of time. So we've got the cnidarians here. Down here, so we've got deuterostomes, and here we have the protostomes, the ecdysozoans, and so the bryozoans, they fit into the lophotrochozoans. So as you said, they should be with the other lophophorates, the brachiopods and the phoronids. So this is historically they were grouped in the Lophophorata. Now as people started to sequence stuff, they were being pulled apart. And even in some more recent transcriptome analyses, bryozoans sometimes group with enteropods and cycliophorans in the polyzoa, they call that group. But they are also, recent analyses, genome-wide analysis, which do recover the Lophophorata. So they do sometimes come together. So it's still, this is why this summary tree is quite recent from 2023. It still doesn't show them together, because there's still occasionally you get a study that says, oh, where they're not together. So it's still some controversy. I think they should be together with the other lophophorates.
Yeah, that was like my other question, if the lophophore is good synapomorphy or not, but you already answered that. That it's still a little bit.
My view, yes. Yes.
Thank you.
Thanks.
Thanks so much. That was mind-blowing. I'm very curious. You said that some of the organisms, they manage to live in the cooling system of nuclear. Actually, working. Sorry. In that actually they can manage to survive like above average radiation levels, or?
Oh, that's a good question. Presumably, I don't know. I don't know how much radiation is in the cooling water, because presumably they can't just. Can they just release highly reactive?
I don't know. I'm just wondering.
That I don't know whether they. Yeah, that's an interesting angle. Whether they are resilient to radiation. They certainly occur there, and with other fouling organisms like hydroids and stuff like that. Yeah.
Thanks.
Okay.
Oh, is this working? Hi. Yeah. Hi, I'm here. Yeah.
Oh, hello.
Thank you for your talk. It's beautiful, and it is nice to have so many fossils in your study group. But even though you have a ton of fossils, I wonder, in that graph that you show us with the extinction and origination rates, right? You talk about a lag, and you highlight that. You mention it, right? There is a lag. But that lag,
yes.
the lag,
Yeah.
It could also be caused by just not finding enough fossils, because it's really old at that point and very hard to find, right?
Yes, of course. In more recent rocks, you find more fossils.
Exactly.
I'm taking the word of my paleontology colleague here, because he knows the fossil record so well, and I kind of trust him. Similarly. If going back to that other time calibrated tree, which has a really
exactly. Sorry for interrupting you, but then you said there, kind of proof what I was thinking, right? In that tree, you said that it shows that the group is older than the fossils.
Yes,
exactly.
So this is when my colleague Paul saw that tree, he almost fell off his chair, because he knows the fossil record so well. Because he also because the cyclostomes, they would occur in the same kind of habitats as the chylostomes, which is the group that we're talking about here. And he would find cyclostomes, but no chylostomes. So he said it's not possible that they were there, because he couldn't see them, because he saw the cyclostomes and they would occur together. So so this is where, I mean, obviously, yes, the fossil record is gappy,
right?
But but I also trust his expertise and estimates there.
Thank you. I have another question.
Okay.
It caught my attention when you talk about the convergence in the skeleton current. Excuse me. And then you talk about, and then, I had this question: what is the source of natural selection in animals such as this, right? But then you talk about abiotic and biotic factors, right? Competition, predation. But what about the reproductive strategies? Do they match the convergence patterns of the skeletal? Like, is there a relation there?
The reproductive, you mean in terms of whether they're brooders or zygote spawners?
Right. I don't study this.
No, I don't think so. I don't think reproduction has an effect. So the examples that I gave for the convergent evolution of all the morphological traits, they all brood. They all, and what they do, they reproduce via polyembryony. So where you have these big brood chambers. So you have a primary embryo that's sexually produced. And then within that brood chamber, this primary embryo gives rise to secondary embryos. And they're all genetically identical in that. And they all do the same. And so I don't think that has an effect on that.
So it would be very hard to tease apart what are the forces driving that convergence evolution?
Yeah, I wouldn't know how to tackle that, to be honest. Yeah.
Thank you.
Thank you. I was wondering, given that perhaps some of the morphological characteristics are arbitrarily picked to create these taxa, how has that informed the morphological matrix that you're doing, or has that caused problems?
Very good question. That is clearly so this is where this matrix, So it will include some homoplasies. It was actually while we were coding, we would, go through the characters, and there was the sound of homoplasy. As we would come towards a particular character, there was this, what should we do here? That because the matrix is across the whole order of the chyllostomes. So it's a family level matrix, across all the families. So you are likely to encounter problems there. We're just seeing this. Okay, we have to start somewhere with this, and of course you can identify the homoplasies as you're running analyses, but it's a problem that we are aware of, and I'm kind of hoping by producing this matrix, I don't see this as a finished product. Even though when we publish it, it will be finished, kind of. But I can, I imagine maybe people will take that matrix and expand it or modify it further. And the other thing is with the matrix. So there was a picture of several people, I don't know, in the room when you saw that. So we basically locked ourselves in a room for a month. We coded, and then we gave the matrix to our Brazilian colleague, and he said, no. So he's modified it further, and it's a very iterative process. So that's the other thing, you because there's more than 400 species in there, and then you add another taxon, and then you have another, hang on, but this has a character state that we haven't accounted for yet. So then you have to add another character state, and then you have to look at all the other 400 taxa and make sure everything is still okay. So it's,
Thank you very much.
Yeah, thanks. Yes.
Well, thank you, Andrea, for your amazing talk. There are so many things that make me very, very, very excited, because you're looking at evolutionary processes acting over a very long time. And even though I would like to delve on morphological matrices and the fossil record in terms of representation along the rocks, because it's not just as you go back in time, it declines. No, there are periods that for different reasons have a better rock record. Exactly. So it's more complicated than simply going back in time. But what I really wanted to get your opinion about is disparity in form over long periods of time. Mm-hmm. And we have these hypotheses that early in the evolution of lineages, mm-hmm. as they appear for the first time, they show a wide variety of body forms. And as evolution goes on, natural selection, extinctions go on, this becomes kind of like skimmed in a way that certain forms prevail, mm-hmm. and major body forms disappear in evolution. Yeah. And the diversity that we have nowadays, it's basically the expansion of one particular body form or very few body forms. So it's more variations on the same topic.
Yes.
Do you see that?
I wouldn't say so. I think because in terms of the different frontal wall shields, we see them still today. And I don't think that's a case. I mean, it's an interesting way to look at it, but I don't think we see that. Yeah, maybe let me think about this a bit more.
Yeah, I mean that's a kind of like a proposal, a hypothesis.
Yeah.
But for example, in flower structure,
yeah,
we don't see. I mean, we see the expansion.
Yeah.
But we don't see the disappearance of ancestral structures.
Yeah, no, but we still see the old morphologies.
So they prevail.
Yeah, they still. Yeah. I mean, they're not. Maybe not. They're not as abundant. So in terms of diversity, they're not as. Exactly. But they're still there.
Yeah.
Yeah. And the thing is because they are modular animals. So this is where it seems that bryozoans can switch between major morphological differences very easily. And I wonder whether this is because they are modular. There's something about, they can switch a gene on and off quickly and produce something. I mean, I don't have it in this talk. There's an example of a species which was described as 3 species before. And morphologically, it includes very divergent morphologies. And they found that it's actually in one specimen. So it's actually one species. So there's something in the switching or in the gene expression that major morphological changes can happen like that. But yeah, it's in a very bryozoans, they are fascinating, but they also give us a lot of headaches. Because we don't really understand. We don't understand what generates this morphology and the polymorphism and all of that. Yeah.
You say that the study of bryozoans are neglected.
Yeah.
Why?
I know you like bryozoans, you told me.
No, but for everybody.
It's I think they're difficult to work with in terms of identifying them. So even if you have ecological surveys, it just says bryozoa, if you're lucky. They're cryptic sometimes. I mean, I've shown you some bigger colonies, but so for example, we went collecting on the Great Barrier Reef, and we would collect. So because on the Great Barrier Reef, yes, you have the corals, but still. But we would pick up coral rubble, bring it back, and you have, a big piece of coral rubble like that. On the underside, you would have like 50 species of bryozoans, and there would be tiny colonies, like a patchwork. But you have to know what to look for, and they're not obvious. They're not in your face. Yeah.
Thank you.
Hi. Thanks for amazing talk. I was amazed. My question is really small. You show us that you have several mitochondrial gene arrangements. My question is, does that arrangements have any phylogenetical signal?
So that's something we still need to look at. I looked at that in one particular group where I did a breakpoint analysis. And it wasn't that informative, to be honest, but I think it's definitely something that we also still want to look at. Definitely.
Yeah, I mean, in the context of this
Absolutely.
huge phylogeny is based on large genomic scale data sets.
Absolutely, yeah, that's definitely.
Okay,
yeah,
thank you.
Wonderful. And also, not just in terms of a phylogenetic signal in the gene order, but also in terms of what are the processes that cause these genome rearrangements. So I think the most common one that people seem to accept is tandem duplication random loss. So, because we have such a diversity of gene order rearrangement, it might be good. It might be a good playing field to test some of those hypotheses and see what are the processes for that.
Thank you again, Professor Waeschenbach. Now we have a coffee break, so see you in 20 minutes. And